Okay, let's finish up chapter 15. So we have transcribed, made a copy of the DNA to RNA. So we are here. We took the DNA, we made a copy into RNA. Now, we're going to take the RNA and it needs to be processed before it can leave the nucleus. If I gave you a copy of something important, I would want you to maybe laminate it or put it in a folder. I've seen your backpacks. I don't want it to leave my room unless it's in a folder or it's processed in some way. So we're making an important copy from the special part of the library where the architect is making a copy, and we need to process that mRNA, put a cap and a tail on it, get rid of the introns. So this is only exons that exit the nucleus, and then we can export it. Okay, so this chapter is just getting us to the point where the mRNA can leave the nucleus. We've made the copy, we've made the photocopy, now it needs to leave the nucleus. So in between there, we are also going to look at the exons, introns, and how that is all spliced, and how also tRNAs and rRNAs are processed. Okay, let's get started, shall we? So after transcription, this is eukaryotic mRNA. Because remember, in prokaryotes, it's all just happening all together because there's no nucleus. So ideally, we're going to take this pre-RNA and we're going to make this delightful mRNA that's going to be ready to leave the nucleus. The exons, those are the ones that are going to exit the nucleus, right? These are the exons. And they are sort of interrupted or intermingled with these introns, which are non-coding. They don't code for anything because they don't leave the nucleus. So we have to remove these. That's step number one. We're removing the introns. They are coded in an RNA stabilizing protein. They protect the pre-RNA from degradation while it is processed and exported. The three most important steps are that protecting it, the addition of stabilizing signaling factors at the 5' cap, the 3' tail, and the removal of the introns. That feels like a test question, right? Doesn't it? What are the three most important steps of pre-RNA processing? Pre-RNAs are, oh, we already said that. Okay. In rare cases, the mRNA transcript can be edited after this if there is some kind of mutation. Sometimes it can actually be edited, or there's alternative splicing, there's different expressions of editing. Sometimes the tRNA structure can be changed. So there is possibilities at this step for things to be fixed or mutated. Let's cap that thing first, right? Let's put on a cap. We're just putting on a cap. 3 at the 5' end, we're going to put a cap, a methyl guanosine cap. So anyone want to guess what that is? It's going to be here. There's going to be methyl groups. There's going to be guanosine there. And it's just a cap. It's just a 5' cap. And what it does is it's just going to, like a hat would do for you, it protects this mRNA as it moves into the nucleus. It protects it from being degraded and this coding part from being ripped off or lost. So that's what the 5' cap is. Again, it's protecting it as it enters the nucleus. There are factors involved in protein synthesis that actually recognize this code and put a cap on it. And it's usually also the cap helps initiate translation. So not only is it important to leave the nucleus, it's also important, as we see in all of the factors, that will initiate translation. Just like we had factors in transcription factors, we will have translation factors. And this 5' cap is one of those. On the other end, we've got a poly A tail. You should not miss this as a multiple choice. What nucleotide are we going to use? It's A. It's a bunch of A's. Look at this. A. We're going to put a bunch of A's on there. About 200 A's. And that's called the poly mini A tail. Goes at the end. It further protects degradation. It's also the binding site for protein necessary for exporting the mRNA to the cytoplasm. Okay, so we've capped it, we've put a tail on it, and now we've got to cut out all of these pesky introns that are going to stay inside our nucleus. They're not going to come out. So the exons are what exit, and the introns stay in. They are removed during processing. They are not coded for. So if we have something like this, we're going to take these guys out, okay? So this is actually pre-RNA or nuclear RNA, and this is mRNA. We will say it a lot that you go from DNA to mRNA, but there's like an intermediary process here. The regions correspond to regulatory sequences, and the biological significance of having many introns or having very long introns is sort of unclear. There's a lot of research still happening into why do we have these introns? What do they do? What do they mean? Why do we have them? What's going on? So the process by which we snip them out, cut them out, is called splicing. And it is done by spliceosomes. That's crazy. I love the names. So the intron itself is non-coding. The beginning and the end of each intron, see, here's our intron. The beginning and the end is actually marked with a specific nucleotide, GU at the 5' and AU at the 3', so that it knows where to cut. Okay? The splicing of the pre-mRNAs is conducted by complexes called spliceosomes. The spliceosome does the splicing. Okay, so it's going to come in and it's going to curl it around, and it is going to splice out what it does not want. See how these come together, and it's going to, then this is going to attach to this, it's going to move it out, and then those are going to attach to each other. Okay? We're doing the same thing with tRNAs and rRNAs, those that make up the ribosome and those that make up the tRNA. Same thing. They are, even though those two are not translated, there's still a lot of processing that goes in once they're coded. They're transcribed, then processed in the nucleus, then released into the cytoplasm to get ready for translation. So here's the rRNA being processed. This is going to end up being that ribosome. We will talk about the ribosome when we get to translation in the next chapter. Mature tRNAs. So the tRNA, this is the codon. This is the mRNA. This part of the anticodon is going to read the codon, and it's going to attach a amino acid so that we will get a long translation peptide chain. Okay? The tRNA folds into position and has an anticodon on one end. The anticodon is not against codons. It's like no codons here. It is for codons, if you will. It's a 3 nucleotide sequence that interacts with the mRNA codon, and it has complementary base pairing, so the 3 line up like this, and it facilitates the addition of a polypeptide chain. We will talk about that in the next chapter. Mature RNA makes up approximately 50% of each ribosome. So the ribosome is going to be that subunit that comes and translates. Okay, so essentially this is a really short chapter where we're just going from mRNA and we are processing it so it can leave the nucleus. Here is what that looks like. Well, maybe. Here is what it should. There it goes.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green.
This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear riboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles.
So I hope that was helpful. I will see you in the next video.